All right. Good evening, everyone. It's still Jane Nnenna here, and today I want to discuss on a very special topic, immunology. Okay, so before we delve into this topic, I just want to make a kind of disclaimer because if this is going to be your first time of hearing about immunology and everything we are going to discuss today, it's actually going to be a kind of confusing, or you may not be able to get everything because there are a lot of things that are interrelated. The explanation will just be looking like, oh, it's just what you explained in the previous one. It's still what you're trying to explain. But I can assure you, maybe after the recording, after the class, you can still listen to it again. I hope it's going to um assimilate you be able to assimilate better. Okay, so if this is your first time, but if you have heard of immunology or if you've done it in undergraduate, please don't just will be as a recap or trying to remind you what you already know. All right, so it's a very interesting topic and I hope you just enjoy it. Okay, so as usual, if at any point you cannot hear me, just let me know so that um we'll get to fix the problem and I'll not talk to myself throughout the whole class. I hope you can still hear me at this point before I start. Please let me know. Can you hear me?
Yes, we can.
Yes,
That's
I
good.
can.
Yes, that's beautiful. So let's get started immediately. So we're talking of immunology. What is immunology? So immunology is the study of the immune system. I know immune system, they help to protect our body from foreign agents or invading organisms, and then our body responses to these foreign agents. These foreign agents can be viruses, they can be bacteria, they can be protozoa, they can be fungi. So the molecules and the cells responsible for immunity constitute the immune system. Okay, so they are different cells, different molecules that help us to fight against these organisms. And then the collective and coordinated response to these foreign substances is what we call the immune response. So the major function of the immune system, like I explained, is to prevent or limit infection by pathogenic microorganisms. So they help to get it removed or help to fight it so that our body will not be invaded. All right. So the recognition of microorganisms and foreign substances is the first event in immune responses of a host. Example, our first line of defense against foreign organisms is our barrier tissues, just like the skin that stop their entry. So the skin is a kind of a first line of defense. So they help to stop a lot of foreign agents from entering into our body. And when they actually make it through the skin, we also have other cells, other molecules inside the body that also help to fight. So the general function of our immune system or immunology we are going to look at today is just to help us to fight against diseases or against foreign substances. So I just want to lay this foundation or introduction so that when we are discussing the subsequent subtopics, we get to know where we are driving at. Okay, so there are two mechanisms of our body defense against all these organisms. Number one, we have what we call the innate or the natural or non-specific immunity. Okay, and then we have acquired, adaptive, or specific immunity. So, what are the innate immunity? They are the resistance that an individual possesses right from birth, and they are the first line of defense against foreign organisms. So, you can just say these are the immunity that one possesses. Maybe you inherited it from your parents or just from the genetic point of view. Okay, so that the natural ones, maybe as a newborn baby, when a baby is born, though the immunity is still low, but they still have a kind of some level of immunity that maybe they get from birth, maybe from their mother through mother to child transmission. This will help them to fight a lot of foreign organisms. Then we have what we call also acquired adaptive or specific immunity. So these are the ones that they help in protection of the body, and the body develops this set of immunity over time, either from exposure to the specific pathogens or germs, and they are the second line of defense. So these acquired immunity, they are the ones that the body have, or the one that the body tend to acquire, maybe due to experience. For example, maybe you're affected with measles. Okay, it's your first time of the measles affecting you. You know, when that measles affects your body, the body will build immunity against that measles, which is a pathogen or an antigen. Okay, I will explain further. Then the next one that this measles will come at to attack you, the body will fight against it and you not come down with measles. You know that most people that have measles, maybe when you're small, you notice that you not have that measles again. Why? Because the body already have what you call acquired immunity. That's the body develops this immunity because this pathogen enters the body and they tend to build that immunity against it. So anytime it comes again, the body will just fight it off and you not come down with it. Okay. So this thing we're just trying to expatiate on the differences between innate immunity and then acquired immunity. Like I said, the innate is the first line of defense that is present from birth, while the acquired or adaptive immunity, they are the ones that developed after exposure to that particular antigen or that particular foreign organisms. Okay, so the innate immunity, they are non-specific, meaning that they respond the same way to all pathogens, why the acquired immunity are highly specific. That means they have a target specific pathogens. Okay, so we also have the response time of this immunity. The innate immunity responds immediately. It takes minutes to hours, while the acquired or adaptive immunity, they are delayed. It takes like days to weeks because the body is still trying to build or trying to study this foreign pathogen. But the innate ones, they're already there from birth. They just start attacking or start fighting for you. All right. So in innate, there's no memory. That response does not improve with repeated exposure. While the acquired immunity, it has memory. Response is faster and stronger on re-exposure, just like the measles case study I gave earlier. All right. So we have the components of the innate immunity. We have the physical barriers like skin. We have the phagocytes, the natural killer cells, the complement proteins. Then for the acquired, we have the B cells, the T cells, and then the antibodies. Okay. So for effectiveness, the innate immunity are less effective in clearing infections alone. Why the acquired or adaptive immunity, they are more effective and they are more tailored to the specific pathogen because this is built up after that particular pathogen has come. So the body will build antibody that is very specific to that particular pathogen. That's why they are more effective. Okay. So like we say, the innate is present right from birth, while the acquired are developed after birth, maybe due to experience. All right. So let's go back to antigen-antibody reactions. So what are antigens? Antigens, they are molecule, often a protein, that triggers an immune response when it enters the body. Okay, so it's essentially a foreign substance that the immune system recognizes as needing to be neutralized or eliminated. So the antigens, they are just any foreign objects that stimulate or triggers an immune response. So what are antibodies? They are also a protein that is being produced by the immune system to specifically bind and neutralize a corresponding antigen. So antibodies, they are globulin proteins called immunoglobulins that are synthesized in serum. That's in the serum is a part of blood, okay, and then tissue fluids, which react specifically with the antigen that stimulated their production. So what is this trying to explain? It's explaining that antigens are the trigger that activates the immune system, while antibodies are the defense mechanism that is being produced to either combat those antigens to by elimination, fighting them, removing them, or trying to reduce them. Okay, so antigens are the trigger, while the antibodies are produced to fight them. That's the defense mechanism. Okay. So the interaction between antigens and antibody are known as antigen-antibody reactions. And it is fundamental to know how the body defends itself against pathogens and other harmful substances. So the reactions are highly specific. That's the reaction between antigens and antibody are highly specific. And then an antigen reacts only with antibodies produced by itself or with closely related antigens. Okay, so Since these reactions are essentially specific, they have been used in many diagnostic tests for detection of the either antigen or the antibody in vitro. There's in vitro studies, like studies carried out outside the body of an organism, like tissue cultures, cell cultures, like all these microbial cultures that is being used in diagnosis, that's in laboratories. Okay, so and also the antigen antibody also form the basis of immunity against microbial disease in vivo, like the clinical trials, researches, studies, animal studies, and a lot of it. So the antigen antibody reactions, they are very, very important in, let's say, in public health. Okay, so what are the nature of antigen antibody reactions? So this nature is trying to explain maybe the properties, the characteristics of antigen antibody reactions. Number one, we have what we call the lock and key model. So it describes the specific and precise binding between an antibody and its corresponding antigen. So this interaction is crucial, that is very important for the immune system's ability to recognize and neutralize foreign invaders like viruses or bacteria. So what this lock and key model is trying to tell us is getting a picture of the key, lock and key. So if you're talking about lock and key model, you have to have a picture or illustration like a key, where the antibody is being regarded like a lock, okay, and then the antigen is being regarded as a key, okay. So what happened is that an um an epitope, which is a specific region of an antigen that an antibody recognizes and binds to, okay, is it like if an antibody is specific to a particular antigen, they will tend to bind together in a kind of padlock, okay. So why the antibody is a padlock, then the antigen is a key. So if they're specific to each other, they will bind together. Okay, but if they're not specific, they will not bind together. There will not be any antigen antibody reaction. So that's what he's talking about. So antibodies produced by the immune system have specific binding sites called the paratopes that complement the shape and chemical structure of specific regions on antigen called the epitopes. So I was trying to explain that epitope is is a part of an antigen which the antibody recognizes and binds to. Okay. So, and then the region of antibody that binds to the epitope is called the paratope. I'm just trying to explain the terms I mentioned with the above explanation. Okay. So the complementary fit is analogous to a key, which is regarded as the antigen, and fitting into a specific lock, which is an antibody. So that's what the lock and key model is talking about. That if an antigen is specific to a particular antibody, they're going to kind of lock with each other, which will create an antigen antibody reaction, okay, or antigen antibody complexes. So this is a kind of a structure. It's just trying to show us the structure of an antibody. So antibody has the kind of Y structure. So when it is specific to an antigen, they're going to bind together using this lock and key model. I was trying to find a particular picture or particular graphic that illustrates this. I was not able to. Maybe if I see, I'll post it on the group. Okay. So let's go back to another property of antigen antibody reaction. They are non-covalent bonds. The bonds that hold the antigen to the antibody combining site are all non-covalent in nature. What are non-covalent bonds? It's a chemical bond that doesn't involve sharing of electrons, unlike the covalent bonds. So they arise from weaker or more dispersed electromagnetic interactions between molecules or within a molecule. So they include like hydrogen bonds, electrostatic bonds, van der Waals forces, hydrophobic bonds. So if you're conversant with chemistry, I think this is going to be, you get to know all these things I'm talking about. Okay, so these are just non-covalent bonds. I just want you to note that the bonds that hold antigen to an antibody, they are called non-covalent bonds. Okay, so another property or nature of antigen antibody reaction is reversibility. They are reversible since antigen antibody reactions occur via non-covalent bonds, which are weaker bonds. They are hereby nature reversible, which means it can be prevented or it can be dissociated by higher ionic strength or extreme pH. Okay, so they can be reversed. So we have another one which is affinity and avidity. So what is affinity? Affinity. So antibody affinity is the strength of the reaction between a single antigenic determinant and a single combining site on the antibody. So it is intensity of attraction between an antigen and antibody. Okay, so affinity is talking about one antigen plus one antibody. That is a single antigenic determinant and a single combining site on the antibody. So high affinity binding is believed to result from a close fit or a close relationship between the antigen binding site and the corresponding antigenic determinants that facilitate development of strong non-covalent interactions between them. Okay, I'm going to show you a picture to explain more on this. Why avidity is a measure of the overall strength of binding of an antigen with many antigenic determinants and multivalent antibodies. So avidity is more than the sum of individual affinities. So the major difference between affinity and avidity is that affinity is just involve one antigen plus one antibody, while avidity involves plenty or two or more antigens and two or more antibodies. Like you can see in the picture below, this is a typical affinity example, which involves like antigen epitope plus one particular antibody, while in avidity, this is an antibody plus two antigens. Then this is also an avidity, which involve like two or more. This one is four antigens plus more than um two antigens. Sorry, the yellow one, like the one that is in Y shape, like I say, is an antibody. Okay, so why the one that is in blue form is the antigen. So the avidity is more than one antibody and more than one antigens. Do you understand why affinity is one antigen plus one antibody? So this should just be the basic. Forget about all the grammar I've been speaking. So we also have another one, specificity and cross-reactivity. What is specificity? Is the ability of an individual antibody combining site to react with only one antigenic determinant, or the ability of a population of antibody molecules to react with only one antigen. So if to say that an antibody is very specific, it means that it has only one antigen that is combining two. Okay, for example, let's say a man is faithful to his wife. It means that it's only his wife that you know is not cheating or is not going to another women. So that's what specificity is talking about. An antibody or a group population of antibody molecules that are reacting to only one antigen is very specific to it. Okay, and in how antibodies can actually show a high degree of specificity to a particular antigen is if number one, if it get to know distinguish the primary structure of that particular antigen, if you get to know the secondary and tertiary structure of that particular antigen, and then the isomeric forms of that particular antigen. Isomeric forms that they are just like different forms that the antigen can exist. Okay, so we have another one, cross reactivity. It's just the opposite of the specificity, where is the ability of an individual antibody combination site to react with more than one antigenic determinant, or the ability of a population of antibody molecules to react with more than one antigen. So you can see they are just different. For example, here, this is anti-A antibody. Okay, AB means antibody, while AG means antigen. Okay, this is anti-A antibody that's in kind of yellow color or orange color. Then it is very specific to antigen A. Okay, this is the epitope of the antigen A. It's very specific to it. It binds to it. But when it comes to cross reaction, this same anti-A antibody will also be binding to antigen B because they have maybe a kind of shared epitope. Like you say, epitope is that part of antigen where the antibody binds to. Maybe they have antigen B have the same epitope with antigen A. So this anti-A antibody will bind to it. Okay, that's a cross reaction. Then we also have another scenario where anti-A antibody will bind to antigen C because they have a similar epitope. It's not the same epitope, but they have a similar. You can see here they have this epitope is kind of smaller or is different from the major anti-A antigen A epitope. Okay, So I just want to show you the difference between specificity and cross reaction. Okay, so let's go back to another one. Factors affecting measurements of antigen antibody reactions. Okay, so get to know that the only way that one knows that an antigen antibody reaction has occurred is to have some means of directly or indirectly detecting the complexes formed between the antigen antibody. So the factors you can use to know if antigen antibody reactions has really occurred is number one, affinity. Like I explained, the higher the affinity of the antibody for the antigen, the more stable will be the interaction, and it will be more easier for one to detect if that interaction has actually occurred. So the second factor is avidity. So reaction between multivalent, that is more than one antigens, and multivalent antibody are more stable and they are easier to detect. Okay. So another factor is antigen to antibody ratio, which is talking about the it influences the detection of the antigen antibody complexes because the size of the complexes formed is related to the concentration of the antigen or the antibody. If you look at the diagram by right hand side, you notice the first diagram have antibody excess, while the second one has antigen excess. The antibody is the one in the orange color, while the antigen is the one in the blue color. Okay, so but in the last diagram have equivalence. They kind of the antibody and the antigen, they are like equally bound to each other, forming what we call lattice formation. So when there is more stable antigen antibody complexes formed, then it will be easier to detect the complexes or the concentration. Okay, so we also have another factor, which is the form that the antigen exists, that's the physical form of the antigen. So if the antigen is particulate. Particulate if it's in solid or insoluble form. One can generally look for agglutination, that's clumping of the antigen by the antibody. So you can easily detect if there's antigen-antibody reactions. But if the antigen is soluble, one can generally look for the precipitation of the antigen after the production of large insoluble antibody complexes. Okay, so these are the four major factors that affecting measurement of antigen antibody reactions. So let's go back to the major tests for antigen antibody reactions. What are different tests we can use in knowing if this antigen antibody reaction has really occurred? Okay, so this test for antigen antibody is majorly useful in diagnosis, especially in laboratory. We're going to be seeing some of the applications. Okay, so we have four major tests, and it's not limited to this. We have a lot of them, but we're going to be concentrating on these four major tests for antigen antibody reactions. We have the agglutination or hema agglutination test. We have the precipitation test. We have the radioimmunoassay, which is RIA, or enzyme linked immunosorbent assay, which is ELISA in short. We also have what we call the complement fixation test. Okay, so there's four tests we're going to be looking at, and we're going to be discussing each and every one of them one by one. So let's talk about what agglutination or hema agglutination test is all about. So it's an antigen-antibody reaction in which a particulate antigen, and what did I say is a particulate antigen? They are insoluble antigen, they are solid antigens, okay? Combines with its antibody at a stable environment, resulting in formation of visible clumping of particles. So this clumping of particles is what we call agglutination. This can be seen with like with a naked eye. You can just Google on your phone what is agglutination or how agglutination is being formed or this clumping of particles, okay? So you can easily see a pictorial view of what we're talking about. So agglutination occurs optimally when antigens and antibodies react in equivalent proportions, okay? And the term agglutinin is used to describe antibodies that agglutinate particulate or antigens. So particulate antigens are antigens that are insoluble or exist in solid or aggregated form. You have an example like microbial cell walls, red blood cells, or the viral capsid. So when the antigen is an erythrocyte, the term is called hemagglutination. So hema means blood. Whenever we hear hem, it means blood. Okay. So when the antigen is used for when the particular antigen that is used for antigen antibody reaction is erythrocyte, for example, red blood cells. So that's when we call it hemagglutination. But when it is other things like the viral capsid or the microbial cell wall, it's called normal agglutination. So I just want you to know the difference between the normal agglutination and then the hemagglutination. Hemagglutination is majorly when it involves blood, okay, like red blood cells, like erythrocytes, okay. We have types of agglutination tests. We have the quantitative and the qualitative agglutination tests. So what are the quantitative agglutination tests? They are the test that is used to determine whether an antigen or antibody is present in a sample. So the presence of a target substance, it can be antigen you are targeting, it can be antibody, links to clumping, that is agglutination of the test sample, indicating a positive result. Okay, so how this is done is that the antibody is mixed with the particulate antigen. Okay, the antibody of maybe a patient's or a human antibody, a human blood is mixed with a particular antigen, and a positive test is indicated by when you see an agglutination, you know, oh, this antigen is actually specific, or this antibody is actually specific to this antigen. That means it is positive. For example, a patient's red blood cell can be mixed with antibody to a blood group antigen, okay, to determine a person's blood type. Do you understand? A patient to determine somebody's blood type, if the person's red blood cells, okay, can be mixed with the antibody to a blood group antigen. So if there is a visible agglutination or agglutination that is clumping between them, it means that the that person's blood type, okay, is very specific to that antigen. Do you understand? So it can be used to determine if someone is in blood type A or AB or ABO or O positive, all those blood groups. So another one is qualitative agglutination test. So it measures the concentration of antibodies against a specific antigen by determining the highest dilution of a sample that still causes agglutination or clumping of particulate antigen like red blood cells or bacteria. So this is done usually by creating serial dilutions of the sample and adding it to a fixed amount of the particulate antigen. So the highest dilution that causes visible agglutination is called the titer and represent the antibody concentration. So what this is talking about is, in as much as the quantitative, like we explained, just measure if there's antigen or if there's no antigen present in the sample. But the qualitative is talking about, okay, if this antibody is present, at what concentration or at what quantity it is, or the amount of the antibody that is against a specific antigen. Okay, I'm going to be showing you a diagrammatic example of what I'm talking about, because I know this may be sounding so similar. But don't worry, like I said, if you have not heard of this before, it's really going to sound maybe kind of confusing, but maybe you watching this again or assimilating it more will really help. Okay, so let me explain what is a proson effect. So a proson effect is observed when the concentration of antibody is high, that is lower dilutions, and there is no agglutination. And then as the sample is diluted, agglutination occurs. So this is just trying to explain, maybe you're trying to form an antigen antibody reactor to detect if an antigen is specific to this antibody. Okay. So you actually dilute it, or when you actually mix this antigen antibody together, there may not be any agglutination. And it does not mean that they are not specific. It just means that they can be the antibody can be so high. Okay, so it can be so high because the antibody is in excess, resulting in a very small complexes that do not clump. But when there is a dilution of this antibody, okay, when you dilute it to a more stable complexes that it can combine to this antigen, then there will now be agglutination. Okay, so that's what a proton effect is talking about. So the qualitative agglutination test is just you combining the antibody, which is the one in the Y shape, plus antigen. So if they're specific, they'll just form a complex that is the antigen antibody complex, as you can see here. So the qualitative agglutination is just to show you if the antibody or the antigen is present. But in quantitative agglutination test, there's going to be a kind of serial dilution, okay, in the sense that you're going to, let's say for instance, in this picture or in this diagram here, we have patient 1, patient 2, patient 3, up to patient 8. Okay, so we have a known number of the antibody in a kind of, let's say in a microtube or in a tighter plate. Okay, so you just added the known number. Maybe here is the highest concentration. Okay, that is 1/2 is the highest concentration, and you then dilute it maybe by adding a diluent, that is water or saline water. You then dilute it to 1/4. You put the same amount of antibody, but is more diluted, just like you adding, maybe you have 5 cubes of sugar, then you add half cup of water, then the next cup you add full cup of water, the next one you have like quarter cup of, so it's just like diluted, and the more you dilute it, the less concentrated it will be, right? So we have the dilution 1/2 to up to 1 over 1024 dilution. So if you want to check if that antibody is specific to that particular antigen, or where they form that particular titer, that they form that they form agglutination, that is clumping, that's where you're going to check. That's quantitative. Okay, so what you're going to do is the you just add the antigen to, maybe to, let's take for instance this patient 1, the first dilution, which is highly concentrated, you add the dilution, there's no clumping because of the proton effect, there's high amount of antibody. So the second one you add again, there's no agglutination. The third one, the fourth one, this up to this 1/64. Okay, you do not see any agglutination. Then in 1/128, that's when you see agglutination. This one, there is a kind of clumping. Okay, that's where you see agglutination. So the titer value is what they tell you. The one preceding the one that has agglutination is called the positive. While the one that actually has that agglutination is what's called the negative. You can see it's this one that my mouse is pointing at. Okay, so we're going to say that the titer value here is then the 1/64. That's the last one proceeding before the agglutination. Do you understand? So that's what this quantitative agglutination is. Help us to dictate. I don't know, have you done a malaria test and they tell you that the titer value is maybe, let's say, 1/4 or 1/8 or so it depends, like the load of the agglutination, like what I wrote here in the application. I wrote a 4-fold rise in titer is generally taken as a significant rise in antibody titer. So when you have a kind of 4-fold rise, like here we have a dilution of 1/2, 1/4, 1 over, so a 4-fold, like that is from 1/16. We consider that, oh, maybe you have antibody that is being produced against malaria. That means you must have malaria infection in your body. So these are just what we use in the lab. And I don't want it to sound like a jargon, okay, or more complicated. These are everyday use, especially with someone that works in a lab, is very conversant with all this. So what are the applications of the agglutination test? Number one, it can be used for blood typing and then transfusion compatibility. So maybe for determining the blood types, like I explained, like the ABO or the rhesus factor to ensure safe blood transfusion. So you can carry out an agglutination test to check if that particular blood is compatible to the one you want to transfuse. Okay, so the clumping of red blood cell agglutination indicate that the donor and recipients are incompatible. You cannot transfuse. Maybe when you mix the blood of the one a patient that you want to transfuse the blood, you mix his or her blood with the blood of the one you want to transfuse into him. Okay, so when the antibody of that particular patient form agglutination with the one you want to transfuse into it, that means they are not compatible together. If you transfuse them, they will cause what we call a hemolytic um a kind of hemolysis, okay, as lysis of the blood. So we also use agglutination tests in diagnosing infectious disease. Okay, they can be used to detect the presence of specific antigens or antibody associated with infection. For example, where the latest agglutination test can identify bacterial antigens in body fluids, for example, for Haemophilus influenza type B. So we also use it in research applications. It can be used to study immune response to various antigen during an experiment. So like I said, this is not a rocket science or something, or it's just a theory. It's something that is used in day-to-day life, especially in the health sector, like in a hospital or maybe in a research lab. So let's talk about hemagglutination. No, I explained that hemagglutination is when the antigen involved is an erythrocyte, as a red blood cell. So they are serological tests that detect the clumping or agglutination of red blood cells in response to an antigen-antibody reaction. So they are used for various purposes, including blood typing, viral titer determination, and then antibody detection. Okay, we have different types of hemagglutination tests. We have the passive hemagglutination test. We have what we call the Coombs test. We also have hemagglutination inhibition test. So let's talk about the passive hemagglutination test. So they involve coating of red blood cells, which is the erythrocytes, with a soluble antigen, and then use it in an agglutination test for antibody to the soluble antigen. Okay, I don't want this to sound so confusing. So normally the agglutination test only works with particulate antigen that's insoluble or solid antigens. But it is possible to coat erythrocyte with a soluble antigen, example viral antigen or polysaccharide or a haptin, and then use the coated red blood cells in an agglutination test for antibody. So this is just like a kind of performing agglutination test, okay, like we kind of convert precipitation test. We're going to explain what precipitation test to agglutination test. So precipitation test is the one that deals with soluble antigens, okay, while agglutination test deals with insoluble or particulate antigens. So if because agglutination tests they are more easier to perform and they're also more sensitive or easier to dictate. So you can actually try to um detect soluble antigens using agglutination tests by coating of red blood cells. You know, red blood cells, they are particular cells with soluble antigen that maybe of target that you want to use, and then you can run agglutination tests with it. So the application include detection of antibody to soluble antigens and the detection of antibodies to viral antigens. So that's what the passive hemoagglutination test is all about. For example, in the picture here, you see an antibody plus an antigen that is being coated. This red blood cell is coated with a soluble antigen. Okay, so when you then put them together, that's when you then have antigen antibody reactions. Okay, because on a normal experiment, you cannot use soluble antigen for agglutination test. Okay, but when you put this soluble antigen on a particulate antigen that is insoluble or solid antigen like the red blood cells, then you can then use it for to help you to easy because it's easier to perform, it's very visible and then easier to detect than when you use only the precipitation test. That's why what's what the passive hemagglutination test is talking about. So we have what we call the Coombs test. So the Coombs test is a blood test used to detect antibodies against red blood cells. So we have the direct Coombs test, we have the indirect Coombs test. So the direct Coombs test is used to detect the presence of non-agglutinating antibodies on red blood cells. That's what the direct comp says, okay? So when antibodies bind to erythrocytes, whenever you hear erythrocytes, just know we are talking about red blood cells. So when antibodies bind to red blood cells, they do not always result in agglutination. They're called the non-agglutinating antibodies, and this can result from the antigen or antibody ratio being in antigen excess or antibody excess, or in some cases, electrical charges on the red blood cells, preventing the effective cross-linking of the cells. So these antibodies that bind to or do not cause agglutination of red blood cells are sometimes called incomplete antibodies. So, and this does not mean that these antibodies are different in structure. Okay, so it just means that they may be like something that is causing it not to agglutinate. Okay, so the direct home test is used to detect the presence of non-agglutinating antibodies. Like for instance, if you combine an antigen and antibody and it's not agglutinating, okay, so this can result maybe if there's excess antibody or if there's excess antigen, or in some cases maybe there are electrical charges on the red blood cell that is preventing it from agglutinating or from forming a complex. So what they do is that you add what we call a second antibody that is directed against the targeted antibody. Just like you adding another antibody that you made it to cling to that antibody, okay, to that non-agglutinating antibody so that it can be able to form a complex. So let me show you a picture of what I'm talking about. I think this is where I actually explain it. So this is the patient's red blood cells. Okay. So when you combine these patient's red blood cells with antibodies and they are not combining or you are not noticing an agglutination. So when you do this Combs reagent, that's anti-globulin, when you add it to the complex, you'll be able to form agglutination that is plugging together. So even though there is an antigen excess or antibody excess, this Combs reagent will actually help it to form an agglutination. So that's what the direct Combs test is talking about. So it's just you adding another extra antibody that will help the incomplete antibody or the non-agglutinating antibody to help them to firsten the agglutination with the antigens. Okay, so we have the indirect home test. So it is used to know whether a serum sample has antibodies directed against a particular red blood cell, and you also want to be sure that you also detect potential non-agglutinating antibody in the sample. So the indirect is just like opposite of the direct. Okay. So in this method, instead of adding that second antibody directly to the antibody, okay, so you just have to, okay, let me just read the method. It's done by incubating the red blood cells with the serum sample, okay, the red blood cells plus the sample, the serum sample. Then when you wash out the unbound antibodies, then adding a second anti-immunoglobulin reagent to cross-link the cells. So a sample of the patient's blood, it can be a plasma or a serum, is taken. Then it mixed with reagents, that is the red blood cells. So if antibodies is present in that patient's blood, they will bind to the reagent's red blood cells. So I don't want this direct and indirect cocktails to actually confuse you. But what I'm trying to explain is this. In the direct context or in the indirect context, the first step is that you're going to add the patient's serum plus the target red blood cell, which is the antigen. Okay, so they could form a complex. So after forming the complex, the 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 complex you then add this Combs reagent, this anti-immunoglobulin, they will then form to help it to agglutinate. So, okay, so when they agglutinate, that's when you're going to be seeing a clog particles. Why in the direct one, you just add the antibody, you just add the Combs reagent directly, okay? You add it directly. You don't have to like form all those complexes before. I don't think this picture is doing justice to what I'm trying to explain to you, okay? So the just For the addition of a second antibody directed against the immunoglobulin antibody that is coating the red blood cell. So this anti-immunoglobulin can now cross-link the red blood cells and result in agglutination. Okay, so that's the major difference between the direct home test and then the indirect home test. So indirect one, the test is done by incubating the red blood cells and the serum. Then you wash out the excess, that is the unbound antibodies, and then you add a second anti-immunoglobulin reagent to cross-link the cells. So a sample of the blood, the patient's blood is taken. So this is just like another explanation. Is taken and mixed with the reagents as a red blood cell. So if antibodies are present in the patient red blood cell, they will bind to the reagent red blood cell. So application that where we can use these home tests is in detection of rhesus factor antibodies. So antibodies to the rhesus factor generally do not agglutinate red blood cells. Okay, so the antibodies to the rhesus factor did not agglutinate. They don't agglutinate red blood cells, maybe due to excess, like I explained, excess antibody or excess antigen, or due to charges on the red blood cells. They will not agglutinate. But when you add a Combs reagent um to this complex, when you add a Combs reagent to the red blood cell, they will then be able to agglutinate. They will cross-link this red blood cell and they'll be able to agglutinate. Okay? Like, for example, the red blood cell from a rhesus positive child born to a rhesus positive mother who has anti-rhesus antibody will be coated with these antibodies. So what this is trying to just explain is that, a rhesus um like a rhesus negative woman that is married to a rhesus positive man. Okay, so when they actually meet and give birth to a child, the child will be rhesus negative. Okay, so if the child is rhesus negative, it means that the mother will then have antibodies of this rhesus factor. Okay, they will have the antibodies, and if the woman get pregnant again, the child may not survive because these are the antibodies will then be fighting the red blood cells of the second child. The first child may escape through, but the subsequent pregnancy will not escape through. So is this home test that is used to test for this, maybe in a lab, is it home test that's going to be used to testing for this to check if the mother already developed an antibodies against the child. Do you understand? So I think this is a more practical example of what I want to explain here. So we have hemagglutination inhibition test, which is another type of hemagglutination test. So when hemagglutination are inhibited or stopped by antibodies specifically directed against the virus, this phenomenon is called hemagglutination inhibition. So it's a serological assay that is used to measure antibody levels against certain viral antigens, particularly, most especially the influenza virus. Okay, so this forms the basis of the viral hemagglutination inhibition test, which is used to detect antibodies in patient sera that neutralizes the agglutinating viruses. So this hemagglutination test is majorly used mostly for the influenza virus to check if person have been affected with this influenza virus. So it relies on the principle that antibodies can inhibit the ability of a virus to agglutinate or clump the red blood cells. Okay, so it's a very simple, sensitive, and relatively inexpensive test. So that's why it's majorly a common method to assessing antibody responses to viral infection. So how is this hemagglutination inhibition test done? First of all, you have to, maybe for the person that you want to diagnose, the patient's serum is first incubated with a viral preparation, that is the antigen of this influenza virus, for example. Then red blood cells that the virus is known to agglutinate are added to the mixture. Okay, the red blood cell that the virus is added to the mixture. First of all, when you incubate this patient's serum, that's the blood of this patient, okay? The blood of the person that you want to diagnose. When you incubate it with the viral preparation, if that viral antigen if the virus inside the incubation is specific to that patient's serum, that means it is a positive test. That means the person actually has an influenza virus. So in the on the second note, for you to confirm if the person actually have, you add another red blood cells that the virus is known to agglutinate. Okay, that you have another red blood cell that the virus is known to agglutinate. So if the antibody is present, this will combine with the viral particles and prevent agglutination. And a lack of agglutination indicates presence of antibody in the patient's serum. So what it's trying to explain is inhibition in the sense that this viral, maybe the virus antigen, you combine it with a serum of the person that is battling with this influenza. When you combine them together, okay, so the combination, they will a kind of form complex, that's antigen antibody complex, right? So another, for you to make sure that okay, this diagnosis is positive or negative, another red blood cells that the virus is known to agglutinate, maybe a red blood cell that this particular virus, that's the influenza virus, is known to cause clumping with, is added to the mixture. So when you add it to the mixture, if the viral antigen does not agglutinate this red blood cell you added, that means the test is positive. That means it has already formed a complex with the serum of that patient. Already formed a complex together. Okay, that it then means that the answer is positive when there is no agglutination between this viral antigen with the second added red blood cells. Okay, that without red blood cells you've already added again. So, but if you add this red blood cell and there's a visible agglutination, it means that the viral antigen did not really combine or did not form a complex to the patient's serum. That means the person is a negative. Okay, so that's what is talking about. So we have the second antibody anti-reaction test, which is called the precipitation test. So a precipitation reaction occurs when a soluble antigen and antibody combine to form an insoluble visible solid called precipitate. Like I said, agglutination test. One thing you have to note between agglutination and precipitation test is agglutination deals with insoluble or particulate antigens. That is insoluble antigens. While precipitation is talking about soluble antigens. Okay, so even if you don't get anything I've been talking about this, I think you should note this key difference between the two tests. So there are different types of precipitation tests. Excuse me, let me take water. So there are different types of this precipitation test. We have the radial immunodiffusion test, which is called the Masseny test. It is a method for quantifying antigen and it is done in an agar gel. Okay, so it involves placing a sample containing an antigen into a well with an agar gel that already contains antibodies against that antigen. So the first keyword you have to know when you're talking about radio immunodiffusion is the diffusion. Diffusion is movement of a molecule from a place of higher concentration to a place of lower concentration, right? So how it is done, just take a look at the picture that this map is pointing. How it is done is that this is an agar gel, okay? So antibodies will be coated in this well. These are kind of wells, like small holes, kind of, okay? So they're already antibodies in these um wells, okay? So antigen that you want to detect, okay? The antigen you want to maybe check if is targeted to the antibody is being placed at each of the well. Each of these well, we have like up to 12 here. So antibody is placed on each of them. So each anyone that there is precipitation, okay, so it involves placing a sample containing an antigen into a well within an agar gel that already contains antibodies against that antigen. So the antigen then diffuses into the gel where it encounters the antibodies. So a visible precipitation ring forms. So the diameter of the ring is directly proportional to the concentration of antigen in the sample. So this just explain that if the antibody and antigen, if they are specific to each other, they will form what we call a precipitation ring, okay, inside the gel, and that's what you're going to use to measure. So we have another type of precipitation test which is called immunoelectrophoresis. So this electrophoresis, you know that we're talking about electric charge electrophoresis. So it's a combination of immunodiffusion and electrophoresis. So it's a method which is different antigens in serum are separated according to their charge, order, and electric field. So in this method, a drop of antigen is placed into a well in an agar on a glass slide. So an electric current is then passed through the agar. You can see the positive and the negative charge here. So when this electric charge is passed through the agar, the antigen move in the electric field according to the charge and the size. So following electrophoresis, a trough, a kind of a cut is made. You can see where a trough is made, where the antibody is placed. Okay, so a trough is cut into the agar and is filled with antibody, and diffusion is allowed to occur. So as the antigen and antibody diffuse towards each other, they form a series of lines of precipitation. So the main advantage of electrophoresis is that a number of antigen can be identified in serum. There's a method is used to detect normal as well as abnormal protein, such as myeloma protein in human serum. So what immunoelectrophoresis is talking about is addition of immunodiffusion, the one we explained previously, and then electrophoresis, which is applying electric charge to immunodiffusion. So when you get a slide is just like a flat surface. Okay, you place an agar gel on it and add antigens, maybe create a well on it and add antigen, maybe at one point of the well. Okay, so you apply an electric charge and you allow this antigen to move according to the charges as well as the sizes. So you then create a well just below a trough, which is like a kind of pot just below the antigens. So you add your antibody, like the target antibodies. So there will be diffusion of this antibody into the well, and when it get to meet with the antigens, they will form what we call lines or precipitations. Okay, so that's what the immunoelectrophoresis is talking about: addition of electrical charges to diffusion. So we have another one, counter current electrophoresis. So in this case, the antigen and the antibody are placed in wells, pounced out of an agar gel, and then the antigen and the antibody are electrophosized into each other, where they form a precipitation line. So this text only works in condition where the antigen and antibody have opposite charges. For example, from the picture, you can see the antigen has a negative charge and then the antibody has a positive charge. So this counter electrophoresis is just using the phenomenon that unlike like poles attract and then the like poles reject. So if they have different charges, they're likely going to come together, and if they form a precipitation, that means they are specific to each other. So it's primarily qualitative, although from the thickness of the band, you can get some measure of the quantity. So its major advantage is the speed. It's very fast to perform. That is a counter current electrophoresis. Okay, so it's different from the normal electrophoresis we explained, that is immunoelectrophoresis, because this one it involves the use of negative and the positive antigen or antibody. So all the use of positive and negative charges, while in immunoelectrophoresis you can just use only one particular charge. So we have the third type of antigen antibody test, which is called the radioimmunoassay or the enzyme linked immunosorbent assay, which is called ELISA. So ELISA are those tests that are based on the measurements of an enzymatic reaction associated with immune complexes. So in any particular assay, the enzyme may be linked to either the antigen or the antibody. So this ELISA is when an enzyme is actually linked to antigen-antibody reaction. So this enzyme it actually helps to detect, or it actually helps to for easy detection of this antigen-antibody reaction. Remember, the focus is to detect different things for detection of antigen-antibody reaction. So this enzyme-linked immunosorbent assays is explaining when an enzyme. So this enzyme, they can be alkaline phosphatase, phosphoradase, peroxidase, just giving different type of enzyme that can be used for this enzyme link that can be used to link these antigen antibody complexes to detect if there is antigen antibody reaction. Okay, so when radioisotopes instead of enzyme are used to label to be conjugated with antigens or antibody, they are called the radioimmunoassay. So this is the difference between radioimmunoassay and enzyme immunoassay. In radioimmunoassay, radioisotopes are used, while in enzyme linked, the enzyme are used as labels, okay, to this antigen antibody complex. So we have different types of radioimmunoassay or the enzyme-linked immunosorbent assay. We have the competitive and then the non-competitive. So when it comes to competitive ELISA or competitive RIA, is used for the estimation of antibodies present in a specimen, such as serum. So just like the name, the principle of the test is that two antibodies are conjugated with enzyme and the other present in the test serum. That is, if the test serum with positive antibodies are used. So there's competition between two antibodies for the same antigen. That's why it's called competitive ELISA. So appearance of color indicates a negative test, that is absence of the antibodies, while the absence of color indicates a positive test, that is presence of antibodies. Okay, so let's use a case study to understand more. In this, let's take for we want to test for HIV antigen if a particular antibody is specific to a HIV antigen. For example, you collected a patient sample, a serum to be tested. They are added to the to a wells, that is the microtiter wells, and incubated at 7 degrees Celsius, and they are then washed. Okay, so this serum, this patient serum, let's say somebody that want to test if they have a HIV antigen, that is if they have the HIV virus. You combine the person's blood, that's the serum part of the blood, with the HIV antigen and maybe incubate them for some time. Okay, so when you wash off that particular, after some time, if the antibodies are present in the test serum, antigen antibody reaction occurs. Like I always say, if they are specific to each other, there'll be a reaction occurs. Then the antigen antibody reaction is detected by adding another enzyme label specific HIV antibodies. Do you understand where I'm going to? So there are two antibodies that are used. The one that contains the serum, okay, that contains the serum of the patient, and then the one that is being labeled or conjugated enzymes, okay, the enzyme label specific HIV bodies. So in a positive test, no antigen is left for the second antibodies to act. Yes. So I want us to understand this particular one, this ELISA, because it involves is a very simple test to perform. Okay, so where let me just recap. You want to test if someone has a HIV virus. Okay, so you collect the person's blood, that's the serum part of the blood. You mix it with the HIV antigen. So when you mix it, after some time, you wash it off. Okay, and then add another enzyme. This enzyme is very specific to that HIV, like it means that it binds to that HIV antigen. So because in a person's blood, it contains antibodies, right? So if the person's blood contains antibodies, that is specific to HIV. That is, if the person is HIV positive, it means that the person's antibody will bind to that viral antigen that they were incubated together, right? So, but if the person does not have any HIV antigen, it means that the antigen will not bind. They will not bind together. They will not form antigen-antibody complex. So the second antibody, which is enzyme linked, that's why I say enzyme linked. Okay, the second antibody is enzyme labeled that is specific to HIV antibodies. So if the person's blood does not contain antibodies against the HIV antigen, that's if the person does not have HIV virus, that means that enzyme liquid will eventually attach to the antigen. Okay, it's eventually attached to the antigen. So appearance of color indicates a negative test, that is absence of antibodies, while the absence of color indicates a positive test. So in a positive test, it means that there's no antigen left for the second antibody, that is the enzyme-labeled antibodies to act. Hence, the antibodies remain free and are washed away during the process of washing. So when substrate is added, no enzyme is available to act. Therefore, positive results indicate no color reaction. Okay, why in a negative test, that is in which no antibodies are present in the serum, antigen in the coated ways are available to combine with the enzyme linked antibodies, and then the enzyme acts on the substrate to produce a color. So let me just lay this foundation. The main purpose of this enzyme labeled or enzyme linked antibodies is for them to, if they actually combine with that particular antigen, they produce a color. Okay, they are linked to the particular color. They produce a kind of a color. Okay, that is a visible color. You can see, oh, this enzyme has been linked to the antigen. That means the test is negative. I don't know if you understand what I'm saying. So a competitive ELISA is most commonly used for detection of HIV antibodies in serum in patients with HIV. So if you don't understand, we'll come back to it again, but let's explain the non-competitive. So they also use the measurement of the antigens and antibodies. So unlike the competitive, where the target antigen competes with a labeled form, that's with a labeled antibody, to a limited number of antibodies or antigen. So the non-competitive assay utilizes excess amounts of antibodies to measure the occupied binding sites. So how does the non-competitive happen? So it's very different from the competitive. You know, the competitive, the antigen and the patient's serum is added, and then another antibody is added again to check if the antigen and antibody, if they actually form a complex. But in this one, it's different. Okay, so the antibody is coated and immobilized onto wells of microtiter, and then the test sample, which is the containing the suspected antigen, is added to the wells and is added to react with the antibodies in the wells. So after some time, then you wash it off, okay? Then a second enzyme conjugated antibody specific for a different epitope of the antigen is added and allowed to incubate. So after removing any free secondary antibody by rewashing, the specific substrate is added and then ensuring homogenic reaction, that is production of a visible color. Okay, so I think a picture will do more justice to it. So this is a competitive ELISA, and then there's non-competitive ELISA. So in this solid phase, when you want for the competitive, which is for antigen detection. So this antigen is the antigen is combined with an antibody, like I explained for that HIV. So the HIV antigen is combined with maybe someone that is suspected to have HIV antigen combined together. Okay. So a labeled antigen, okay, is then that is the enzyme link, that is the one on blue, is then added again. So if this antigen, if the antigen and then the antibody, sorry, if the antigen and antibody do not form a complex, okay, this one that is labeled, that is enzyme link, will then form a complex with the antigen. And then it will show color, which is showing that, okay, it's negative. But if they already form a complex with this antigen, with this antibody, so with the patient antibody, that means when they add the second one, it will not form any complex, okay? It will form a color with the antigen. Do you understand? So both of these antibody, this orange color Y and the blue color Y, they are both antibodies. They are competing for this antigen. Okay, so if this antibody is specific to the antigen, they will form a complex, and then when the second one is added, you do not see any antigen to bind to. That means there will not be any color to form because they're the one that is labeled and carry a particular color. So let's go to the non-competitive. So when the non-competitive, the antibody, okay, is the one that is being attached with the antigen, maybe antigen in a patient sample. So the antibody are immobilized in maybe in a microtiter plate, and then the target antigen are added. Okay, so if they actually form a complex, another antibody that is targeted to another epitope or another part of the antigen is being added again. Do you understand? So this one that is not competing with the antibody is only going to attach to another part of the antigen. Okay, so if they form a complex, this one will actually attach to it. That's the labeled one, and then it will produce a color. Do you understand what I'm talking about? So the chromogenic reaction is then compared with itself. That's for the non-competitive. In a positive test, an enzyme acts on the substrate to produce a color, and its intensity can be measured by spectrophotometer or ELISA reader. So the change in color can also be observed by the naked eye. So these are the different applications of the non-competitive ELISA. So let's go back to the fourth antigen-antibody reaction test. We have the complement fixation test. So this complement fixation is a process where complement proteins bind to antigen-antibody complexes, marking them for destruction by the immune system, thus leading to the elimination of the pathogens and foreign substances. So, and what is the complement protein? They are is a system of plasma proteins that work together with other immune system components to eliminate foreign agents. So these complement protein, they are part of the immune system. Okay. So what actually happens is that when you want to carry out a complement fixation test, it helps to detect specific antibodies in a patient's serum. So the complement fixation test is that an antigen is mixed with the test serum that is to be analyzed for antibody, and then the antigen-antibody complexes are allowed to form. So if there is presence of these complement proteins, if there's no antigen complexes that is present in the tube, there's none of the complement will be fixed. But if there's antigen-antibody complexes present, there will be a fixed complement, and thereby reducing the amount of complement in the tube. What this is talking about is that, like I explained, these complements, they are part of the immune system, okay? They're part of the plasma proteins that help to fight against diseases. So what happens is that when you actually mix a particular antigen and antibody, okay, when you want to detect if there's the presence of a specific antibodies in a patient's serum. Okay, so if the antigen and antibody that you mix together, if they form what we call a complex, okay, that means this complement will also be fixed because they help in fighting against foreign objects. So if they form complex, if this antigen and antibody form a complex, that means the complement will be fixed within. Okay, so if they do not form a complex, that means the complement will not be there. The complement proteins will not be fixed. So after allowing the complement fixation by any antigen antibody complexes, the standard amount of red blood cells which have been pre-coated with anti-erythrocyte antibodies is added. So the amount of the antibody-coated red blood cells is predetermined to be just enough to completely use up the complements initially added, so if it were used. So if all the complements are still present, that's if there's no antigen antibody complexes formed, that means the antigen that's between the antigen and then antibody in question, that means all the red blood cells will be lysed. Do you understand? But if the complement fixation test, if they are formed with the antibody complexes, that means there will not be any complement to cause red blood cells lysis. Do you get? So the complement fixation is talking about ability of maybe when you do an antibody antigen test. Okay, so these complement proteins, which are part of the immune system, they will be fixed. Will be fixed in the sense that they will attach to the antigen antibody complex that is already formed. That's if that antibody is specific to the antigen. But if they are not specific, they will not form any complex. Okay, so when a standard amount of red blood cell which have been pre-coated with anti-erythrocyte antibodies, if they're added, okay, and if there is formation of that antigen complexes, which leads to the formation of complement fixation, okay. So when they are added, that means the 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 additional red blood cells, okay, then they will not be lysed. Okay, that's if all the complements are still present, there's no antigen antibody complexes formed between the antigen and antibody. That means all the red blood cells will be lysed. All right. So, but if all the complements are not, if they are still fixed, okay, that's when the antigen antibody complexes, if they are formed, that means the red blood cells, there will not be any available complement to lyse. So this complement actually causes lysing of the red blood cells. So if they are not fixed, that's when there is no antigen antibody complex. That means all the and all the red blood cells wouldn't be lysed. Okay, so that's what the complement fixation is talking about. Let's say, for example, here you have an antigen, okay, in this patient's serum. This is the patient's serum containing the antibodies, right? So if this antigen if there's antigen present and is specific to the antibody, that means they will form a complement, or they form a complex, sorry, which lead to fixing complement fixation, which leads to fixing of these complement proteins in the body. So when the red blood cells are being added, as a coated red blood cells are being added, that means there will not be any available complement to lyse them. There will not be red blood cell lysis. So what we actually check out for in complement fixation test, if there's lysing of red blood cells. So if there's lysing of red blood cells, that means if there is lysing of red blood cells, that means the antigen is not present. Okay, so in the second picture we have no antigen present, and then in the patient's serum which contains the antibody, so there's no antigen antibody complexes formed because there's no antigen. Okay, so the complements are not fixed. So when you add red blood cell, those red blood cell that is targeted to the antibody, there will be lysing of the red blood cell. Okay, do you understand? That it means that there is no antigen present, number one, and then the complements are not fixed, and then the antibody will then cause lysing of the added red blood cells. Do you get? So that's for the complement fixation test. Then we'll go back to another one, which is called the test for the cell. Sorry, I need to mark this maybe. Okay. Please, at this point, can you hear me? Let me know if I'm not talking. Zabi, can you still hear me at this point?
Yes, I can.
I hope I'm not speaking Javans. Because I know this may sound so weird.
It's quite complete.
Yeah, I get. Okay. I need to shoot this. Just give me a minute. Let me. Okay. So we have another one. Test for cell-associated antigens. Test for cell-associated antigens. I wanted to shoot this. Illustrating my screen. Okay, we have tests for cell-associated antigens. They are majorly called immunophenotyping. So these cell-associated, they involves identifying specific proteins to antigens on the surface of cells, which can be useful for diagnosing various diseases like leukemia or lymphomas. So cell-associated antigens, they are used for identifying antigens. They are cells, okay? Identifying antigens on the surface of various cells, okay, which can be used to either in diagnosing a lot of disease. Maybe you want to check for leukemia if someone is having or suffering from leukemia, so you can check for the cells on the cells on the surface of that particular tissue, okay, if they contain the antigens, that causes the leukemia infection. So that's what the cell-associated antigen is talking about. So there are different techniques or different tests that can be used. We have the immunofluorescence test. So it's a technique where an antibody labeled with a fluorescent molecule, which can be a fluorescein or rhodamine or any of the other fluorescent dyes, is used to detect the presence of an antigen in or on a cell or tissue by the fluorescence emitted by the bound antibody. And so the immunofluorescence is just talking about labeling an antibody with a dye or something that will make it to be more visible, okay? Like something that will make it to produce a color. So when it is mixed with an antigen, so if it is specific, if it's specific for that antigen and they form a complex, they will then show a color, they will then show a visible color telling you that okay, there's presence of that particular antigen. Do you understand? So that's what the immunofluorescence is talking about, is labeling the antibody with a particular fluorescence or a dye that shows emit a color or a light. So there are different types. We have the direct or indirect immunofluorescence. So the direct immunofluorescence, you just add the fluorochrome label, that's the labeled antibody directly to the antigen. Okay, the antigen on the surface of the cell, so you just add it together. So if there's a complex, that is antigen antibody complex, this they will show a color, okay? This will emit a color or a light because it's labeled. You see the antibody is the one in blue color, while the antigen AG is the one in yellow color. So it's added directly. So why the indirect immunofluorescence? It means that the antigen is first of all added with the antibody that is unlabeled is first of all added to the antigen, while the fluorochrome labeled antibody is added later, which is targeted to this antibody is labeled again. Okay, so it's added later, so it's an indirect, meaning that the the labeled antibody is not directly um, added or mixed with the antigen. So that's the difference between the indirect and the direct immunofluorescence. And then the indirect fluorescence is more sensitive than the direct immunofluorescence since there is amplification of the signal due to additional added antibody. Okay, so the only thing you have to know when it comes to immunofluorescence is that the antibody that is being added is used to identify specific antigens on the surface of cells, okay? So which can be used to diagnosing diseases. So the antibody that is being added, they are majorly tagged with fluorochrome dyes, like the fluorescein or rhodamine that emits lights or colors, okay, depending on the color you want. So when there's a complex form, that is the antigen and the antibody, they form a complex, then there will be light or color emission. Okay, we also have another one which is flow cytometry. So this flow cytometry is very common in clinical laboratory to identify and enumerate cells bearing a particular antigen. So the cells in suspension are labeled with a fluorescent tag, just like their immunofluorescence, and the cells are then analyzed on a flow cytometer. So this flow cytometer will help to check to measure the physical and the chemical characteristics of the cells. So the difference between the two of them is that the flow cytometry are being analyzed using a machine called flow cytometer, which helps to check the physical and the chemical characteristics of the cell as they flow in a stream fluid past a laser. So this happening inside the flow cytometer. So you can read up for the flow cytometer provides information on the size, the shape, and the fluorescent particles. Okay, so this is the method it's actually used to analyze these cells. All right, so we have the general characteristics of a specific antibody response. The general characteristics of a specific antibody response. So number one, antibody response, like we said, is specific. So let me start from the first one. Is self or non-self discrimination. Okay, this is the first characteristic. It means that antibodies they distinguish between self and non-self, and it only reacts against non-self, which are the foreign um antigens or the foreign organisms. So they were able to they have a characteristic of recognizing between um their cells and then between the foreign bodies. Okay, so that's one of the characteristics of specific antibody immune response. Then another one, it has a memory. Antibody response has memory. It remembers if it has seen an antigen before and it reacts to secondary exposure to an antigen in a manner different than after a primary exposure. So the more you build like a particular disease affects you, the more it builds memory. The antibodies in your body, it builds a kind of memory that immediately is detect that antigen, it just eliminates it because you already know, oh, this is coming again, or I will not allow it to invade. Okay, so another characteristic is specificity. Okay, so it's a response to a particular antigen is specific for the antigen or a few closely related antigens. So it's very, very specific in the sense that you're able to identify a particular antigen that is specific to. So this was a question that was asked by Chioma in the group. So she was asking to discuss in general the various antigen antibody reaction tests. I think it's from the past question. Okay, so I think from what we've explained today, you will be able to um, discuss the various antigen antibody tests, like the agglutination or hemagglutination test, precipitation test, we have the complementation test, and we have the ELISA or the radioimmunoassay test. So I think from today's discussion, we'll be able to discuss that. Then we also have another topic, which is the fate of immunogen. So it's just a very small topic you can be able to read up from the material. Okay, fate of immunogen. So an immunogen is a substance that is often a protein, is an antigen that elicits an immune response in a host organism. So what happens when an immunogen enters the body? So that's what it's trying to talk about. And I also want you to note the different phases of primary antibody response. They are all in the material, just like 2 or 3 pages. Okay, so you just have to read it all. So in case if question is coming from the fate of the immunogen, but for the antigen antibody reaction, we've done justice to that. Okay, so I think that's it for tonight. And if you have any question, you can ask.
I have like 1 billion questions, but I think I'll just have to watch the video over and over again.
Yes, that's why I started in
Yeah,
the beginning that is going to be kind of confusing.
it is
Okay.
actually very confusing. But I think if I should be able to read
Yeah.
and watch the videos again, I think it will, I don't know, it will stick, hopefully.
Oh, okay. No worry, but
Thank you so
if in
much.
case if you watch it again and you still have questions, I just drop it on the group, okay?
Okay. All
Yeah.
right. Thank you so much for your time. Really appreciate.
Thank you. So thank you for staying through. Okay. So
Yeah.
I think at this juncture, we're going to call it a day.
Night.
Good night.
Okay, have a wonderful night rest.
You too.
Rest well. Bye.
Bye.